Hello everyone. Today we are starting with a completely new topic. We have focused so far on the metabolism of carbohydrates, and we have completed the story on the catabolism and anabolism of carbohydrates. So now we're going to follow the almost similar pattern like we did for carbs, but we're going to shift our focus to lipids, and we're going to start the story throughout the upcoming several videos about the metabolism of lipids. And following the same pattern as for carbs, first we're going to talk about the first stage of catabolism. If you remember, that is degradation of complex molecules to their building bricks. Speaking of lipids, that is going to be degradation of complex lipids to their building bricks, for example, fatty acids or so. And we mentioned that the first stage of catabolism is usually described by the phenomenon of digestion and absorption of biomolecules. So this is going to be our first topic. But just in order to warm up a little bit, I'm going to remind you about the numerous and important functions of lipids in our organism. So the first thing that comes to our mind is definitely that lipids are the largest energy reservoir in our body. And we all know that 3-acylglycerols are actually responsible for formation of energy. So by degradation of 3-acylglycerols and by hydrolysis, further degradation of fatty acids and enrollment of products in the Krebs cycle, we are capable of synthesizing significant amounts of energy, which is going to be our next topic in the next video. Of course, in the part of general biochemistry, we discussed a lot about the structures of different lipids molecules. We discussed it in detail, so now I'm not going to take your time in getting back to the structure of 3-acylglycerols and the rest of the lipid molecules, because I count on you and your knowledge. The second thing that is very important, and I think that we all know about that, is that lipids are structural elements of cell membranes. So we all know that it's the basis of a lipid bilayer structure of a cell membrane, and actually that bilayer is nothing else as two layers of phospholipids oriented specifically. So the polar heads are oriented to the aqueous medium and non-polar tails towards the central part of the bilayer. We all know that in the structure of cell membrane, we have cholesterol and some glycolipids incorporated. Also, lipids are involved in the formation of nervous cells, and they're involved in the impulse transmission. And again, we have phospholipids there and some sphingolipids, which are very characteristic for the nervous tissue. And we have mentioned several types of those molecules in general biochemistry. Let me remind you about a large and important group of hormones, which are called steroid hormones. And they're responsible for the regulation of very important processes in our organism, but they are all synthesized from cholesterol as the main precursor, so they are considered to be lipid molecules. Also, prostaglandins are molecules synthesized from arachidonic acid, which is fatty acid, and they're involved in our immune response. Not to forget lipoprotein or liposoluble vitamins A, D, E, and K. We mentioned at the time in general biochemistry course that those are vitamins with a specific lipid structure, because if you remember, we mentioned that they're all polymers of isoprenoid units. Bile salts are also synthesized from cholesterol, so they are state-of-the-art lipid molecules, and they are actively involved in our today's topic, digestion and absorption of lipids. Plus, they are also involved in the elimination of excess amounts of cholesterol from our body, but that is going to be the topic from one other video and for some other time. Also, lipids are considered to be very good thermal isolators, electrical isolators, and there are many other functions of lipids. So let's start from the beginning. Significant amounts of lipids are taken into our organism through diet. So let's talk about the content and lipid profile in a diet. So there is a data available in your recommended literature and textbooks where it's stated that average human diet implies the following intake of approximately 40% of carbs and 40% of lipids, 12% of proteins, and 8% of other constituents. But these percentages may vary a lot depending on the individual dietary habits, depending on the recommendation of health organization. So for example, in the past few decades, lipids are recognized as the major risk factors for development of atherosclerosis and subsequent cardiovascular diseases and cerebrovascular diseases. So it was recommended to lower lipid intake in diet as much as possible. Nowadays, there are more and more data suggesting that it's not about the lipids, it's about the glucose and inflammation in blood vessels that are actually causing atherosclerosis and accumulation of those lipid plaques on blood vessels. So it's changing all the time, and as the research develops and we have new scientific results, and these ratios are changing. If you're, for example, applying so famous keto diet, you're going to increase the content of lipids in your diet, and you're going to decrease the intake of carbs, and so on. So these percentages may significantly vary, but this is something that is stated as an average intake. So, but more or less, whatever you take and whichever percentage you consider as an accurate one, we can conclude that there is a high content of lipid molecules in an average human diet. Speaking about the numbers in number of grams per day, an average daily intake of lipids ranges from 50 to 100 grams a day. Speaking about the profile, dietary lipid profile, the highest content of 3-acylglycerols are actually found in that lipid intake. 98 to 99% of all lipids, dietary lipids, are actually 3-acylglycerols. Having in mind the structure of 3-acylglycerols, that is going to be like up to 95% of fatty acid, and the rest is glycerol. And just a small amount, only 1 to 2% of all amount of taken lipids in a diet are actually cholesterol, phospholipids, liposoluble vitamins, some other steroid molecules, terpenoid molecules, and so on and so on. Regarding the fact that the major lipid in our diet are 3-acylglycerols, 3-acylglycerols are going to be considered to be the major sources of saturated and unsaturated fatty acids for our organism. Okay, we have to have in mind that our organism can synthesize saturated fatty acids and monounsaturated fatty acids, and we're gonna see the metabolic pathway in the upcoming videos. But what is of essential importance is that actually those 3-acylglycerols are supplying us with essential fatty acids, which are monounsaturated fatty acids like linoleic, linolenic, and according to some authors, arachidonic acid as well, which are essential because our organism cannot synthesize those polyunsaturated fatty acids. Digestion of lipids can be divided or classified in three stages or in three phases. The first stage or the first phase of digestion is so-called intraluminal phase, which is nothing else but the digestion itself. So it means chemical and physical modification of different lipid molecules prior to the absorption process. And we are going to go through this phase in details. The next phase or the next stage is cellular phase, which is nothing else but the absorption phase, which means that those modified molecules, or let's say digested material, now enters the intestinal mucosa cells, enters enterocytes, and we're going to see which mechanisms are involved in such. Also, this phase actually explains how those digested material is remodified or resynthesized to get lipid molecules again, and to get them ready for post-absorptive phase and form. And the last phase is transport phase, which explains how absorbed lipids are further transported from intestinal mucosal cells to peripheral tissues, either by lymph or bloodstream. And we're gonna go into details with this phase, not that much today, but we're gonna dedicate one separate lecture to the transport and phenomenon of the metabolism of lipoprotein particles. But today we're gonna focus on those first two phases, intraluminal phase and cellular phase. So let's start with the first one, which means that we're gonna follow the same patterns as we did for carbs. We're gonna start explaining the events in different parts of gastrointestinal tracts. So we're gonna start with the mouth and the events happening in the mouth. So what is going on in the scope of lipid digestion? Nothing much. So of course we have mechanical shredding of food and mixing with saliva and getting the content ready for further steps of digestion, but actually no chemical changes of lipid molecules occur. In carbs digestion, we had chemical degradation of starch and glycogen by alpha amylase. By speaking of lipids, no chemical changes, no enzymes involved in this stage of digestion. So after swallowing, the content reaches stomach, and there are two things going on at the level of the stomach. First, we have, due to the movement of the stomach and those mild contractions of the stomach, we have some kind of very weak and mild mechanical emulsification and mixing the gastric content with gastric juices and enzymes and hydrochloric acid and so on. But the efficacy of this mixing and emulsification is rather low, which is gonna make the whole phase of lipid digestion rather slowly and weakly efficient. But this at this point, at the point of stomach, we're gonna start the digestion of lipids. So stomach is gonna be the starting point of chemical degradation or hydrolysis of lipid molecules. And we have two enzymes active at this point. First, it's lingual lipase. This enzyme is synthesized in lingual glands, but it's not getting active, it's not activated in saliva, it's activated in the stomach, where we have an acidic pH. You know that very well because of the presence of hydrochloric acid. So this enzyme is so-called acid-resistant lipase, so it's active in acidic medium. And many students are actually confused, and they say that the digestion of lipids starts in the mouth by the action of lingual lipase, but it's not actually the correct statement. So digestion starts in the stomach upon the action of lingual lipase. We have one other enzyme which is called gastric lipase. This enzyme is synthesized by chief cells of stomach mucosa, the same which are responsible for the synthesis and secretion of pepsin. We're going to see that in the metabolism of proteins. But this enzyme is active in acidic medium, but it's more active, much more active at neutral pH. And I'm going to explain why is that going to be important. What is the catalytic function of those two lipases? This is going to be easy. Whenever we say lipase, that means that the enzyme is responsible for hydrolysis of 3-acylglycerols. And that is the case here as well. So 3-acylglycerols are degraded or hydrolyzed to 2-monoacylglycerols and fatty acids. At this stage of digestion, actually 3-acylglycerols containing short and medium chain fatty acids are actually digested. Short and medium chain fatty acids are let me remind you, containing 4 to 12 carbon atoms, and a significant amount of these fatty acids are found in cow's milk. So we can conclude that actually dietary dairy lipids digestion occurs in to the significant extent at the level of the stomach, which is very important in breastfed infants and young children, because the content of dairy lipids and milk lipids in their diet is much higher than in adults. And in infants, especially in breastfed infants and small children, stomach juice is not that acidic, so it's closer to the neutral pH. So the effects and activity of gastric lipase is significant, digesting dairy lipids. In adults, this enzyme loses its importance and loses its activity because of the highly acidic medium found in the stomach. So because of this, some authors even claim that there is no digestion of lipids at the level of stomach, and they claim that the exact digestion starts in duodenum and in duodenal phase of digestion. So we're moving on from stomach. The content is released by opening of pylorus to duodenum, and the critical phase of digestion in duodenum is actually the process of emulsification. So what is going on, and what was the problem of the gastric phase of digestion? The problem was that fats are insoluble in water. So in the stomach juice, they existed in the form of large fat globules. So digestive enzymes can be active only at the surface of those large lipid globules. So at the surface, at the contact surface of the lipids with an aqueous surrounding medium. So in order to increase the efficacy of digestion, we have to do something. What? We have to increase the contact surface with the aqueous surrounding. So we have to break this huge fat globule into a large number of small lipid droplets, which is gonna increase digestion efficacy. This is accomplished by the involvement of two emulsification mechanisms. The first one is mechanical emulsification, which is completed by the intestinal peristaltic actions. So there is peristaltis of our intestinum, and it's actually mixing the content of the duodenum, And if you know that even from elementary school, when you're mixing the mixture of water and oil, you're gonna um, you're gonna break apart those huge fat globules into a large number of those fine fatty oily droplets. But if you leave the mixture to stand still, an immediate aggregation of those small droplets back to those huge fat globules occur. So we have to prevent it. So mechanical emulsification is necessary, requiring, but not enough. So we have to stabilize those small fat droplets by binding of bile salts to them. Because bile salts are surfactants and they exhibit certain detergent activity. So the role of bile salts attached to those small lipid droplets is to stabilize those droplets formed by mechanical emulsification, thus preventing the recomposition back to the large lipid globule. And now, when we have a huge number of those small lipid droplets, we actually increased the contact surface, and we exposed a large amount of lipid molecules to the action of digestive enzymes. So the efficacy of digestion significantly increases. Apart from bile salts, fatty acids can also exhibit certain detergent properties, but those properties are much weaker comparing to the efficacy of bile salts. Also, before I move on to enzymes which are active at this phase of digestion, this duodenal phase of digestion is hormonally regulated phase of digestion. And two hormones are involved in regulation of this stage of digestion. But let me just point out that there are much more digestive hormones involved in regulation of the digestion of biomolecules. But at this very point, we don't have a space or time to go into details, so we're gonna discuss about those digestive hormones later on in a chapter on hormones. So the first hormone which is important to be mentioned here is secretin, which is a peptide hormone. It's secreted and synthesized and secreted by intestinal cells. And the signal for its secretion is low pH of incoming stomach or gastric juice. So actually this hormone acts on pancreas, and slightly it acts on liver as well, to start secreting large amounts of bicarbonate ions, which role is actually gonna be to neutralize or to buffer those hydrogen ions from hydrochloric acid and gastric content, and to increase the pH of duodenal juice to the range from 6 to 8, which is an optimum pH for the activity of numerous duodenal enzymes. I mean pancreatic enzymes active in duodenum. The next hormone which is important to be mentioned here is cholecystokinin or pancreazimin, which is also a peptide hormone, but it's synthesized and secreted by lower duodenal cells and yeyunum cells. And the signal for secretion of this hormone is the presence of food itself, especially the presence of lipids and proteins in the content. So this hormone has numerous points of action. So first it acts on gallbladder, causing its contractions and the release of bile, which we saw is necessary for emulsification. Then it acts on acinar pancreatic cells to release pancreatic enzymes. which are gonna, of course, digest not only lipids but carbs and proteins as well. And the third point of its action is pylorus and stomach, but this time it inhibits the gastric emptying and thus regulating that the content which is going to be processed in duodenum is not too large. So it's like in doses which is regulated by this cholecystokinin. So when all those conditions are set up, we can continue with the digestion of lipids. So at the level of duodenum, one pancreatic lipase is responsible for hydrolysis of 3-acylglycerols to 2-monoacylglycerols and fatty acids, so like we had with lingual lipase and gastric lipase as well. But at this very point, a small percentage of 3-acylglycerols can be degraded completely to fatty acids and glycerol. But it is important to point out that pancreatic lipase cannot hydrolyze 2-monoacylglycerols. So in order to get glycerol and fatty acid, 2-monoacylglycerols has to be isomerized first, and this is represented by the list below. So 3-acylglycerols in the first step are hydrolyzed to 1,2-deacylglycerols, and fatty acid from the position number 3 is released by the action of lipase. Then 1,2-deacylglycerols are further hydrolyzed to 2-monoacylglycerols, again by the action of lipase. Then we said 2-monoacylglycerols has to be isomerized to 1-monoacylglycerols first by the action of isomerase, and then lipase is going to continue the hydrolytic action on 1-monoacylglycerols to convert it to glycerols and fatty acids. So as the final products of this phase of digestion, we're going to get the mixture of approximately 75% of 2-monoacylglycerols, 5% of 1-monoacylglycerols and 1,2-deacylglycerols, and approximately 1/5, 20% of glycerols and fatty acids. Pancreatic lipase is the enzyme which requires the presence of one protein which is called colipase. This protein actually binds to both the enzyme and the lipid molecule to be hydrolyzed. Why is this so important? It's important because if bile pigments bind to the protein, they're gonna inhibit its activity. So binding of colipase is gonna do two things. First, binding of colipase, it's gonna enable the upcoming binding of 3-acylglycerols to the active site. Why is that so important? Because in pancreatic lipase, active site is protected by one part of the molecule. So 3-acylglycerol cannot bind to the active site. But when colipase is attached to the enzyme, it causes certain conformational changes which actually results in the exposure and opening of the active site. At the same time, obtaining some kind of hydrophobic medium so that 3-acylglycerols can be efficiently attached to the active site. At the same time, it prevents the inhibition of enzyme activity caused by the presence of bile salts. There are some other enzymes also active at the level of duodenum, digesting some other lipid molecules. Cholesterol esterase is the one hydrolyzing cholesterol esters, as you can see here, to free cholesterol and fatty acids. And we have a group of phospholipases, 4 enzymes A1, A2, C, and D responsible for hydrolysis of phospholipids. So phospholipase A1 and A2, they are hydrolyzing ester bond at C1 at C2 of glycerol, releasing fatty acids, and we have lysophospholipid as a remnant after removal of fatty acids. Phospholipase A2 is interesting because at the position number 2 of phospholipids, usually arachidonic acid is found. So arachidonic acid, I've already mentioned, is involved in the biosynthesis of prostaglandins, and it's responsible for the adequate immune response of our body. So corticosteroids, both natural and artificial medicines, corticosteroids are actually inhibitors of phospholipase A2, thus preventing the release of arachidonic acid and subsequent inhibition of prostaglandin synthesis and subsequent inhibition of the inflammatory response of the body. Phospholipase C removes the polar part of the molecule, so we have deacylglycerols formed and the polar part of the molecule. And finally, phospholipase D removes the alcoholic group. It's mainly alcoholic, if you remember general biochem course when we discussed the structure of glycerophospholipids. So phosphatidic acid is released. It was believed that phospholipase D is a plant enzyme, but now we very well know that it's found in human organism as well, and thus exhibiting certain regulatory activity. So, as the result of the digestion of lipids, we get the mixture of fatty acid, monoacylglycerols, deacylglycerols, cholesterol, phospholipid digestion products. We have liposoluble vitamin. So all those products now have to be absorbed to enterocytes. But, or however, it looks easy because membrane is of lipid structure. Those are lipid molecules, hydrophobic products, hydrophobic membrane. So one could think that it's going to be so easy for those products of digestion to pass through membranes without any kind of difficulties and the necessities for protein transporters like we had to have for carbs and so on. But no, there is a problem, which is of course solvable. Enterocytes are actually surrounded by a protective so-called unstirred water layer. So it is an aqueous medium surrounding enterocytes, and actually it protects enterocytes from the action of digestive enzymes. So in order to solve the problem and to pass that aqueous barrier, those products of lipid digestion are incorporated in the form of micelles together with bile salts. And this formation of micelles is going to increase the solubility, and actually it's going to enable an easy pass of these molecules through the aqueous layer, which is represented here by the figure. So these are the products of digestion of dietary lipids. So they're packed together with bile salts in the form of micelle. So they are hydrophilic, so they can pass this protective layer, and those products of digestion can be released to enterocytes, and bile salts are brought back to intestinal lumen. So bile salts, we can conclude that they are also of essential importance, not only for digestion, but for the absorption of fats and liposoluble vitamins as well. Those bile salts which are brought back to the lumen after the absorption, they are reabsorbed up to 95% and brought to enterohepatic circulation, and by enterohepatic circulation to liver, where they're going to be directed into the bile gallbladder, and it's gonna, they're gonna be deposited, and they're gonna be ready for the new digestion, for the release, for new emulsification, and for the new absorption process. The importance of this process can be illustrated by one example, which is found in persons and individuals with bile duct obstruction. So if you have an obstruction of bile ducts, you're not going to have an adequate release of bile salts into intestinal lumen. So you're not going to have an efficient digestion of lipids. So emulsification is not going to be is not going to be completed in the right way, and even those lipids which are digested, they cannot be absorbed because there are no bile salts for formation of micelles. So those persons are going to quickly eliminate both undigested and hydrolyzed lipids by the feces, and it is known by the term steatorrhea or fatty feces. Notice that short chain fatty acids do not have to be incorporated in the form of micelles, so they can freely pass the water layer, the enterocytes, and enter directly to bloodstream and brought to liver, and then from liver to peripheral tissues. What happens then? In enterocytes, the resynthesis of dietary lipids occur. So first we have to do something with fatty acids. So we cannot use them in the form we got them upon the digestion. So fatty acids actually form complexes with one specific protein, which is marked as IFABP, or fatty acid binding protein. And there are two things which are actually accomplished by this binding. First, the effective solubility of fatty acid increases, because if you remember, we mentioned that they're not soluble molecules, despite the fact that they have carboxylic group, but due to that long hydrocarbon chain, they are non-polar molecule and not soluble in water. So by binding to this protein, the effective solubility increases. That is the first thing. And the other one is that by binding to this protein, cells are actually protected from the detergent activity of fatty acid salts or soaps. Of course, I'm going to repeat, short and medium chain fatty acids, they can directly enter portal bloodstream, and they are bound to albumin and transported to liver, and then liver is going to distribute those fatty acids either to some biosynthetic process or degradative process, or export them to central circulation and transport them to peripheral tissues. But long chain fatty acids, they undergo TAG resynthesis in enterocytes. So TAG resynthesis in enterocytes occur at the level of smooth endoplasmic reticulum. And first, if you can see from the figure, fatty acids have to be activated in the reaction with ATP, where fatty acid AMP complex is formed, and then coenzyme A is involved in the reaction, and instead of AMP we get fatty acid coenzyme A. This reaction is catalyzed by thiokinase, and this unique activation of fatty acid is going to be also found in beta oxidation of fatty acid and some other metabolic pathways of lipids. So this is not the last time that we're going to talk and discuss about the activation of fatty acids. So thus activated fatty acid coenzyme A may now undergo reaction with 2-monoacylglycerol to give diacylglycerol first, and then the third fatty acid coenzyme A can be incorporated to give resynthesized 3-acylglycerols. Notice, and this is important to remember, that the resynthesis of 3-acylglycerols in enterocytes is different from the one taking place in liver and adipose tissue. So here we saw that the precursor for the resynthesis is 2-monoacylglycerol. In liver and adipose tissue is going to be phosphatidic acid. Apart from the resynthesis of 3-acylglycerol, resynthesis of cholesterol ester and phospholipids also occur, and now all those resynthesized lipid molecules can be incorporated in the form of the first type of lipoprotein particles, which are kilomicrons. If you remember our general biochemistry part, we discussed about those lipoprotein particles, and we mentioned that the main role of the formation of those particles is to overcome the problem of high insolubility of lipids in water. So actually, in order to make them slightly more soluble, those lipid molecules are packed and combined with certain protein molecules which are marked as apolipoprotein molecules, thus increasing the solubility of those particles, and the formation and packing of those particles occur at Golgi apparatus. So smooth endoplasmic reticulum is responsible for the resynthesis of 3-acylglycerol. Rough endoplasmic reticulum is responsible for the synthesis of those apolipoproteins. And Golgi complex actually combines all those synthesized products to form kilomicron. So kilomicron consists of dietary 3-acylglycerols. Knowing the content of TAGs in our diet, it is now logic that 98-99% of the kilomicron content are going to be 3-acylglycerols. The rest of the content are dietary cholesterol, dietary phospholipids, and apolipoproteins. Specific apolipoproteins which are incorporated in the form of kilomicron are ApoA, which are structural apolipoproteins for the maintenance of the proper shape of the particle, and ApoB48, which is very specific for kilomicrons. So the particle is formed, which is marked as nascent kilomicron, and these nascent kilomicron particles are then secreted to lymph and then through thoracic ducts to blood circulation. In blood circulation, nascent kilomicron is going to be remodified to mature kilomicron, undergoing the following degradation or hydrolytic processes, but more about the fate of kilomicron and about the metabolism of some other lipoprotein particles are going to be discussed in the chapter on metabolism of lipoproteins. But so far, with the formation of kilomicrons, we actually completed the digestion and absorption of lipids. So that would be all for today's topic, and I honestly hope that it was understandable, that it wasn't that complicated, and moreover that it was interesting. So until the next topic, which is going to be dedicated to the further degradation of fatty acids, I'm saying bye bye and see you next time.